What comes into your mind when you hear lipids? You might think of fats, oils, grease. But lipids aren't just that. There's so much more. And we're here to show you how important of a role it plays in our daily lives. Get ready to bond with us in this adventure of a lifetime. So before we start the actual discussion, let's first discuss our learning objectives. So by the end of the discussion, we should be able to explain why triglycerides are efficient energy reserves. We should also be able to describe how phospholipids and cholesterol maintain membrane structure and function. Next is we should be able to differentiate major lipid types based on structure and roles. We should also be able to illustrate how lipids acts as signaling molecules and hormones. And we should also be able to analyze health effects of lipid imbalances like hyperlipidemia and atherosclerosis. Lastly, we should also be able to evaluate the impact of lifestyle and hormones on lipid-related disorders.
So for the topic outline, we will begin with what lipids do, including their roles in energy storage and is a part of cell membranes. Next, we will discuss the types of lipids, which will include phosphoglycerides, sphingolipids, and steroids. After that, we will explore the role of lipids in cell communication, focusing on their function as messenger molecules and hormones. And finally, we will look into the problems caused by lipid imbalance, such as too much fat in the blood, genetic disorders, and hormone problems.
Before we dive into the lesson, let's start a quick hands-on experiment that you can do at home using simple materials. First, get a brown paper bag and divide it into two sections. Label one side as oil and the other as water. Now, using a dropper or pipette, place 1 drop of cooking oil on the oil side and 1 drop of water on the other. Let it dry for around 3 to 5 minutes, then hold it up to a light source. What do you see? You'll notice that the oil spot stays translucent, while the water spot either evaporates or disappears. That translucent stain is a positive result for lipids. You can even try this with other food items like peanuts, chips, or cheese. Just crush or rub a bit of it on the paper and test. If it leaves a similar translucent mark, it likely contains lipids. This simple test helps us see how lipids behave differently from other substances. And with that, let's begin exploring what lipids really do in the body, why they matter, and how they keep us alive and functioning. First, let's talk about lipids as an energy source. Lipids, especially triglycerides, are our body's most efficient form of stored energy. They pack more than twice the energy per gram compared to carbohydrates and proteins. Thanks to their long hydrocarbon chains, which release lots of energy when oxidized. What makes them even better for storage is that they're hydrophobic. Unlike glycogen, they don't need water to be stored, which makes them more compact. That's why fats is stored in adipose tissue in animals and in oil bodies in plants. Now, how do we use the stored energy? When we need it, like during fasting or exercise. Triglycerides undergo lipolysis, breaking down into glycerol and free fatty acids. These fatty acids enter the mitochondria, go through beta oxidation, and eventually produce ATP or adenosine triphosphate through the Krebs cycle. And that, in short, is how fat fuels our body. Now moving on to their structural role. Lipids form the foundation of cell membranes, specifically the phospholipid bilayer. The heads are hydrophilic and face water, while the tails are hydrophobic and face inward. This setup helps regulate what goes in and out of the cell. Cholesterol is also a key player here. It acts like a stabilizer, preventing the membrane from becoming too fluid and heat or too rigid and cold. This helps maintain proper cell function and communication. Building on what we've discussed about the structure of the cell membrane, let's now focus on how lipids function as a selective barrier. The membrane's phospholipid bilayer has a hydrophobic core which plays a key role in this function. It blocks polar or charged molecules, preventing them from passing through easily. In contrast, small, nonpolar molecules like oxygen and carbon dioxide can move across freely. This selective permeability is crucial because it allows the membrane to control what enters and exits the cell, helping maintain internal balance or homeostasis. Alright, let's kick things off with a quick brain warm-up. Think through this with me, and I'll give you a moment to answer in your head before I reveal the answer. So which of the following can freely cross the hydrophobic core of the lipid bilayer? Is it A, acetyl-CoA, B, fatty acids, C, sugars, or D, small nonpolar molecules? The correct answer is D, small nonpolar molecules. These tiny, uncharged molecules can slip right through the hydrophobic region of the membrane, kind of like how a fish swims through water. No problem at all. Now that we've looked at how lipids function in energy storage in membranes, let's zoom in on the three major types of lipids and their biomedical roles. These are phosphoglycerides, sphingolipids, and steroids, each playing unique and crucial functions in the body. So first is phosphoglycerides, also called as phospholipids. These are probably the most well-known lipids, especially when we talk about cell membranes. In fact, they're the main component of the lipid bilayer that forms the structure of all our cells. Their design is pretty clever. A hydrophilic head that loves water, and the hydrophobic tails that avoid it. This dual nature lets them create that perfect barrier in the cell membrane, keeping the inside of the cell protected while still allowing communication with the outside environment. But phosphoglycerides do more than just form barriers. They also play a big role in signal transduction, basically helping cells send and receive chemical messages. When a cell is stimulated, some phospholipids can be cleaved to produce secondary messengers like DAG or diacylglycerol and IP3 or inositol triphosphate, which then trigger a cascade of responses inside the cell. Phospholipids are also present in lung surfactant, which helps reduce surface tension in the lungs and prevents the tiny air sacs, which is the alveoli, from collapsing when we exhale, which is very essential for breathing. Another role, they also help form lipoproteins, the transport vehicles that carry cholesterol and other lipids through the bloodstream. Without them, fats wouldn't be able to move around the body efficiently. Next up is sphingolipids. These are structurally different. Instead of a glycerol backbone, they're built on a sphingosine backbone. That might sound like a small change, but it gives them different functions and properties. One key type of sphingolipid is sphingomyelin, which is a critical component of the myelin sheet, the insulating layer that surrounds the nerve fibers. Think of it like the protective coating on electrical wires. Without this insulation, nerve signals would be slow or disrupted. But sphingolipids don't stop there. They are also involved in cell signaling, helping cells communicate, grow, and even self-destruct through apoptosis, or program cell death, which is important for tissue health and development. Fortunately, problems with sphingolipid metabolism can lead to serious diseases. For example, Tay-Sachs disease and Gaucher's disease, which are both genetic conditions where certain sphingolipids build up to toxic levels because the enzymes that are supposed to break them down are missing or malfunctioning. These disorders primarily affect the nervous system and can be life-threatening. Lastly, let's talk about steroids, particularly cholesterol and its derivatives. Cholesterol often gets a bad reputation, but in moderation, it's actually essential. It helps maintain the fluidity and stability of cell membranes, especially in animal cells. Without cholesterol, our membranes would be too rigid or too fluid depending on the temperature. So you could call it like a quality control agent of the membrane. Beyond structural support, cholesterol is also the precursor for several important molecules. Steroid hormones like cortisol, estrogen, testosterone, and progesterone, which regulates many processes from metabolism to reproduction. Vitamin D, which is crucial for bone health and calcium absorption. And lastly, bile acids, which are necessary for digesting dietary fats. But as with anything, balance is key. When cholesterol, especially LDL cholesterol or low-density lipoprotein cholesterol, builds up in the blood, it can contribute to atherosclerosis, a condition where plaques form in arteries, increasing the risk of heart disease and stroke.
Okay, time for something a little more fun. I'll describe something and you try to guess what it is. Plus, I'll give you a scrambled word as a hint. So first is, this lipid acts like a high-tech coating for your neurons, insulating and protecting them so nerve signals move fast. Got it? The answer is sphingomyelin, part of the myelin sheet that wraps around the nerve cells like a protective cable cover. Super important for fast signal transmission.
Lipids are involved in both intracellular signaling, or within the same cell, and intercellular signaling, or between cells. Think of them like the text messages or alerts sent inside the body to control inflammation, immune responses, growth, and even reproduction. There are three major types of lipid messengers. First is we have the eicosanoids, which come from a fatty acid called arachidonic acid. These lipids are short-lived but very powerful in triggering fast responses. There are three main types. First is the prostaglandins. These cause inflammation, pain, and fever. So when you feel body pain or get a fever, prostaglandins are likely involved. Next is we also have the thromboxanes. These help in blood clotting. They are crucial when you get a cut. Lastly, we also have the leukotrienes. These are involved in immune responses and are especially active during asthma attacks. So next time you take a paracetamol or an anti-inflammatory drug, remember, it works by blocking these lipid messengers. Next is we have the sphingolipids. These lipids don't just make up membranes. They also act as second messengers. What does that mean? When a signal hits a cell, sphingolipids help pass on the message inside, especially for responses like cell growth, stress reaction, apoptosis, or again program cell death, like when the body removes damaged or infected cells. So sphingolipids are like middlemen to make sure the cells react properly. Now let's talk about phosphatidylinositol derivatives, especially a molecule called BIP2. BIP2 can split into two important messengers. We have the DAG, or diacylglycerol, and we have the IP3, which is the inositol triphosphate. So these two activate into protein kinase C, which changes protein activity inside a cell, and also calcium ion, which can trigger muscle contraction, enzyme activity, or hormone secretion. So this pathway is like a domino effect that starts with a lipid and ends with a full cell response.
Unlike water-soluble hormones, lipid-soluble hormones can easily diffuse through the plasma membrane because of their non-polar nature. Once inside a cell, the hormone binds to a specific receptor in the cytoplasm, and together they form what's called the receptor-hormone complex. This complex then moves into the nucleus, where it triggers gene transcription, meaning it activates certain genes to produce messenger RNA or mRNA. Finally, this mRNA is translated into proteins, which then go to alter the cell's activity depending on the hormone's function, like regulating metabolism, growth, or development. Now that we're warmed up, let's test your understanding a bit more. So let's answer this question. Which of the following lipid-derived messenger is correctly paired with its function? Is it A, thromboxane,
promotes fever. B. Prostaglandin triggers inflammation and pain. C. Leukotriene assists in blood clotting. Or D. Cortisol regulates blood calcium levels. And the correct answer is B. Prostaglandin. These guys are responsible for things like pain, fever, and inflammation. When you have a headache or a swollen ankle, prostaglandins are often behind it. So after learning how lipids help with cell communication, let's now talk about what happens when things go wrong, when lipid levels go out of balance. This topic is important because lipid imbalance is a major cause of heart disease, stroke, and other health problems. So when we say lipid imbalance, we're referring to too much or too little fat molecules in the blood. The most common problem is hyperlipidemia, meaning high level of fats in the bloodstream, especially LDL or low-density lipoprotein, which is often called bad cholesterol, and also triglycerides, which is also another type of fat that stores energy. Too much of these can clog your arteries, raising your blood pressure and increasing your risk of heart attacks and strokes. And also, there is a less oxygen that reaches the brain or the heart, which is also a risk of stroke or heart attack. So when arteries get blocked, the heart has to work harder, so that will give you a high blood pressure. In hyperlipidemia, excess fat builds up along the artery walls. This condition is called atherosclerosis, where arteries become narrow and stiff. So even though fats are essential, too much of them can be deadly. Some people have genetic conditions that affect their lipid levels, even if they eat healthy. Two examples are familial hypercholesterolemia. This causes very high LDL levels even from childhood. People with this disorder can develop heart disease at an early age. Next is we also have the 𝙤𝙤𝙟 disease. This is a rare condition with very low HDL levels. So HDL is the good cholesterol that removes excess fat from the blood. Without enough HDL, fat just builds up. So yes, even having too little of a good lipid can be very harmful. Did you know that hormonal imbalances can also mess with your lipid levels? Here are some examples. We have the hypothyroidism, which as low thyroid hormone can increase LDL levels. We also have diabetes, often causes high triglycerides and low HDL. We also have estrogen deficiency, especially after menopause. This can lower HDL and increase heart risk. This is why doctors often check lipid profiles when diagnosing or monitoring these kind of conditions. Let's finish strong with some questions that tie lipids into everyday health. Which lipoprotein is considered bad cholesterol because it contributes to plaque buildup in arteries? Is it A, HDL, B, LDL, C, VLDL, or D, chylomicrons? And the answer is B, LDL. Low-density lipoprotein or LDL carries cholesterol to tissues, but too much of it can stick to artery walls, leading to heart disease. Alright, we've covered a lot today, from the vital roles of lipids in body signaling to what happens when lipid levels go out of balance. By now, you've likely absorbed most of the key points, but just to be sure, let's put your understanding to test. So please grab a pen and paper. A few short questions will appear on the screen, and try to answer them as honestly and confidently as you can. No pressure. This is just to help reinforce your learning. So here are the questions. Next, now that you've learned about the essential roles of lipids, it's time to apply what you know, not just in theory, but in real life. This activity will help you reconnect your food choices with your energy levels. So take your time and be honest with yourself. So here's what you're going to do. So first is, you're going to track your food intake for 3 days. So you're going to list all the meals and snacks you eat for 3 consecutive days. You should also highlight the lipid-rich food, like fried dishes, dairy, oils, nuts, butter, any lipid-rich food. If you can, please also note the type of fat, saturated, unsaturated, or trans fat. Next is, you're going to rate your energy. At the end of the day, rate how energized you felt. Is it 1, very low energy? 2, low energy. 3, moderate energy. 4, high energy. And 5, very energized. You can also jot down what might have affected your energy. It could be sleep, stress, exercise, or hydration. Lastly, you're going to write a very short reflection in just 5 to 7 sentences. So it should answer the question, did you notice any patterns between your fat intake and energy levels? Which fat made you feel better or worse? Were there days you felt sluggish or more energized? And why? Has this changed your understanding of lipids in your diet? And finally, what changes do you plan to make, if any? Your reflection can be handwritten or typed. The most important thing, be honest and make it personal.
Before we end, here are the references we use for this discussion. If you'd like to learn more or explore the topic deeper, we highly recommend visiting or reading these sources as well.
And there we have it. To wrap this lecture up, lipids are so much more than just stored fat. They are energy providers, cell membrane builders, chemical messengers, and even disease indicators. Understanding how they function and what happens when they go off balance helps us make smarter choices, not just in classroom, but in life. Thank you so much for listening and participating. This has been your lecturer, Mary Amodia, and I truly hope you carry these insights not just in your notes, but in your lifestyle. Till next time, stay curious and stay healthy. Bye!